the first question, Jenny, is one that you and I have talked about a little bit, and I'll just touch on briefly, is whether there's an R package available for augmented design. And
okay.
I could let you go first. You're
yeah, I'm not aware of that, but you've looked into that more than I have, so why don't you go ahead and answer that question.
Yes, I was going to say, to my knowledge, there is not an R package available for augmented design. One of our future objectives is to develop an R script based on Jenny's presentation today, so that we will make that publicly available to people. As well, at Plant Breeding and Genomics, we have a lot of interest in R, so we would like to share whatever resources are available. And we've got a few more questions here. One question, Jenny, and I'm sure you'll have to qualify this, is what's the minimum required number of checks?
Yeah, that really depends on what crop you're working with, what traits you're working with, how heritable they are. You know, the rule of thumb is that you need to have 10 degrees of freedom for error. So you have to balance your number of blocks and number of checks to you know, get that value of 10. But that's just a minimum value, and that, for example, in corn, I know from having been worked a lot as a corn breeder, you need a certain plot size, minimum plot size, and you also need a certain number of reps to get be able to pick up significant differences among families, say half-sib families or full-sib families, for yield. So I wouldn't, I think even if you had quite a lot of checks, you would potentially not be able to pick up differences for yield in corn with a single rep. So in that case, you know, there's no way to really account for the fact that you don't have multiple observations to estimate your means for the families. So I guess so ballpark would be 10, but that, you know, in some cases that won't be sufficient, and in other cases where you have a highly heritable trait, you know, you almost wouldn't even need to have 10 degrees of freedom.
Great. Thank you, Jenny. And we've had a couple more requests for the link to the presentation, so I've typed that into the chat box.
Okay.
And there's one comment here suggesting that genetic reps in large families may help make up, I believe, make up for some checks, I believe.
Yeah.
Yeah.
Yeah. I think perhaps they're thinking about the case where you'd be looking at taking data on individual plants, and that would be then constituting your family. For Metaphone, we're looking at a plot mean basis. So or else I'm just not quite understanding the comment. Oh. Yeah.
Sorry, okay. So the actual genetic replication in large families. Genetic replicates substitute for technical replicates in detecting your trait differences and increasing your heritability.
Okay.
That's the suggestion.
All right, I'll have to think about that.
Yeah.
Think about that. Again, that may be referring to a case where you're taking measurements on individual plants within each family.
Sure. Yeah, my apologies. I got us on a little bit of a digression there. I apologize. We had another nice comment from someone saying that they know of a package in R called DIGGR for designing P-rep trials.
Well, that would be of interest. That's sort of it's still an augmented design, but basically without the checks, because you're just using some of the replicates. I mean, some of the entries as repeating some of the entries, replicating those so that they act as, they give you the estimate of standard error that you need. I haven't worked with those, but they looked very interesting when I looked at that paper.
So thank you very much for that comment. And another question is, are you aware of other researchers at OSU using augmented experimental design in their breeding work?
At OSU, well, the barley program is the one that I know about. I know, for example, when I was working in Africa, they were using augmented designs in cassava, and that was, in the particular case, they had, there was a cassava mosaic virus epidemic in East Africa, and they needed to move some resistant varieties from West Africa over to East Africa, but the varieties had very different quality and growth properties, the new varieties. And in East Africa, the farming systems were quite different, so they used augmented designs to evaluate potential new introductions in that area. That's one case I know where augmented designs were used very effectively because they weren't limited in seed as one of the reasons that we use augmented designs, but they were wanting to test as many different farming systems and environments as they could. So that was one very nice application of augmented designs. I don't know of any other breeders here who are using augmented designs, but there may be some.
Great, thank you, Jenny. And we've got some more technical questions here for you that you can do your best with. A nice question here, actually. If we are testing a large number of entries at several locations, do we need the same checks across all locations, or can we do some common checks and some checks for specific locations?
I think you could do it. I think you would have options. You're going to get an independent estimate of error within the location, and so that it would kind of make sense to do that. In fact, if you wanted to use additional checks within each location that maybe had specific adaptation to that location, you could use that to get a better adjustment of your block effects there within the environment. But you might have some broadly adapted varieties that you would use across all of your locations, and that would be then a nice way to tie together the information from the different sites. So that would be seems to me to be a very reasonable approach.
Great. And then a question here about your breeding program. What acreage is required for your meadowfoam research?
We're a very small scale program because the current production of meadowfoam in Oregon is only about 5,000 acres. So this is a crop that's been used because of its potential, and the real constraint for us is the markets, developing the markets for the crop. As I said, it's currently used in the cosmetics, and the use of meadowfoam in cosmetic products is gradually increasing. But presently, some of the other industrial applications are not, in practice. So limited acreage. So we have a very small scale project. So I'm usually only, maybe 3 acres or something per year, plus all of my isolated seed increases and so on.
Great. And we've got a nice comment here from the wheat breeding group at the University of Kentucky, and they say that they have been using an augmented design to test yield in early generations when they don't have large quantities of seed.
Right.
And they, yeah.
Yeah, I had in my in the most current version, I had wanted to make that comparison, particularly for, as I said, like in corn, I would be reluctant. I would rather have, say, 200 families with 2 reps per site than 400 families with single replicate, just because I know that I wouldn't have very much chance of picking up detectable differences among my families with just a single rep. But in a crop like either a clonally propagated crop or one, an inbreeding, self-pollinating crop where you're using some sort of pedigree selection system, when you put your initial material out in the field that's just the first go at it. So, for example, in Oregon here, we have a lot of difficulty with stripe rust in barley. So in their early generations, main objective may be simply just to get rid of stripe rust susceptible lines. So, but then they know they're going to advance that material another generation and can do more in-depth screening in subsequent years. So that's really an ideal situation where you'd want to, be able to, in order to be able to test as many genotypes as possible, you might want to use a single rep in the first year of evaluation. So it depends on the crop and also on the heritability of the traits of interest.
Sure. And you got a couple nice thank yous from both grad students in the Moose Lab at Illinois,
Okay, well,
and
you're welcome.
as well from the wheat breeding group in Kentucky.
Okay.
And we have another question here asking about outliers.
Okay.
And wondering if they should be, or does it make sense to remove outliers based on the residuals of unreplicated entries across multiple locations?
It doesn't. I don't know how you would do that because you really only have the residuals estimated for, if you're doing an ANOVA analysis at least, you only have the residuals estimated for the check entries. The new entries don't have residuals. They're all zero. So I guess you'd have to have some other means other than a typical residual plot to figure out what really, was just an improbable observation. Yeah, I really don't know what else to say about that except, if you have something that's completely out of the ballpark, then you may just have to throw it out. But I'm not quite sure what criteria you'd use to decide that.
Mm-hmm. Great. Well, we have a little bit more time for questions if anyone has things that are still pressing. And I see here, I will actually share the links again. Let me try this again.
It may be a good idea also to check and see which version of the presentation was posted.
Yes, and I did check, after we had the mix-up.
Yeah.
For anyone who is still on, I did update the presentation online.
Okay. So if they printed it out before the presentation, they probably had the older version?
Yes, they would want to go back.
Mm-hmm.
And we have one more question here. I don't know if you want to comment specifically in wheat and the number of checks that you may be looking at. We've talked some about how it depends on the trait and the heritability of a trait.
It also would depend on the block size that you want to use too. Different crops, and also if you have some sense of the environments that you're working on, the field heterogeneity that you have to account for. Once you've been working in an area, you kind of get a sense of, well, if I go above, say, 20 entries in this particular crop, this particular site, I'm likely to have too large a block size to get good comparisons within blocks. So it would also depend on, how big your blocks are and how you need to represent, have checks representing that area.
Okay, another thank you here to you, and also a question about using augmented designs for QTL or association analysis. Are you aware of anything in that area?
Well, in the barley cap program, some of the data that we received from the Oregon State breeding program was based on augmented designs. So, and then the objective of the Hordium Toolbox, the database that was developed through that project, was to do association analysis, look for marker trait association. So I guess that would be one example. I don't know specifically of other examples, but that would be one case.
Sure, okay. And you have a thanks from a former OSU barley grad as well,
Okay.
who was able to join us today.
Mm-hmm.
So Okay, here's another possible suggestion when we're looking at number of checks that we may want to use.
Mm-hmm.
That there's a suggestion that this person has read that they thought you should have at least the square root of the number of new entries.
Yeah, I've seen that too. Yeah, I've seen that. I don't, I think in the information from the Indian Agricultural Research Center, I gave reference for their software and so on. That's they do have capability in their little package to help you estimate that using that criterion.
Great. And coming back to the question regarding QTL and association analysis, we have a comment here that fusarium head blight was scored using an augmented design, an association, or QTL analysis.
Right. Mm-hmm.
And another nice comment here that augmented designs help us to increase our genotypic replication, though at the expense of our line replication.
Mm-hmm. So thinking of perhaps like an F2 family to represent have numbers of individuals from that F2 family rather than the individual lines coming from the population.
And another question, could an augmented design be used for selecting lines from wild accessions with small seed amounts compared with checks?
Yeah, I think that's one possibility. In fact, again, with the materials online from the Indian Research Center, I forget the specific name, but they also mentioned that as being a primary use, where you're getting in new accessions from, say, gene bank or something like that, and you don't have much available. So that would be a very appropriate use of augmented designs.
And one comment, or one question that relates back to the design in our field space. The question is, across locations, field size restrictions may apply. How would you take that into consider while you're designing your plots?
So you're saying maybe there at one particular site you have smaller area available than at other sites? Well, I think there wouldn't be, yeah, I think, I'm just trying to think off the top of my head here. Yeah, I suppose if you're going to combine information across sites, it would be desirable to have the same plot size, to have, common variance, but I'm not really entirely sure about that.
Sure, and I would think off the top of my head as well that you would want to maintain, say, the same plant spacing across locations as well, I would imagine.
I do remember from one of Federer's papers that the common variance was not essential, but that's all that I can say.
Sure.
So that there's one reference there, that 2001 paper, I think it is, where they combine information across sites, and that was a comment he had that would not be essential, but I guess I don't quite understand why that is.
Okay, and we have one more question here regarding the square root guide that we mentioned briefly. So if you use the square root guide, is that the number of reps of a check variety or the number of different genotypes?
Yeah, that's a good question. I don't actually know which one that refers to for that particular rule of thumb. I prefer to use the minimum number of degrees of freedom for your ANOVA criterion, in which case you can adjust your numbers of checks and reps, to obtain that minimum number. So I'm not sure for the other criterion which one that refers to.
Sure. And it it looks like we are just about out of time for questions today. And please join me again in thanking Jenny for a great presentation.